So this brings up the question is, why can these be regulated? Is this due to some type of specific structure within these 3' UTRs, or what? So this kind of brought up some new questions that we needed to more directly address. But so far what we saw with this is that it seems that UPF1 and GTP1 are regulating the decay of the mRNAs with highly structured UTRs, and it does appear to be global. But what I can't really say is so far none of this has been based on the 3' UTR. So we kind of wanted to confirm that all this is through the 3' UTR, and to do that we were able to utilize a luciferase assay, in which we have a dual luciferase plasmid in which the renilla luciferase can be tagged with a 3' UTR of interest, and then the firefly luciferase can act as that internal control. And what we could then do is we inserted 2 highly structured UTRs and 2 poorly structured UTRs into this assay for both G3P1 wild type in white and G3P1 knockout in gray. And what you can see is that only when you insert the 2 highly structured UTRs in this plasmid do you see any effects when G3P1 is knocked out, and that you see this upregulation of only the highly structured UTRs. So this is now really showing that the 3' UTR is responsible for this regulation. This system now also gives me a great way to manipulate these 3' UTRs. Plasmids are very easy to manipulate. We can put whatever we want in there and ask some good questions with this assay. So the 3 that I'm going to talk about today are, is this sequence independent? Can I show, is this the sequence that's facilitating this degradation, or is it the overall structure as we thought? And then the other question is if it is structure, can I identify the minimal unit required for this regulation? And then lastly, everything I've been focusing on is the structured region. So now the question is it due to the structured region, or can these non-structured regions play some role in inhibiting this interaction? So first, looking at sequence independent, one easy thing you can do is we simply inserted the 3' UTRs in the reverse complement orientation. By doing so, any sequence-specific features are now lost by being in the reverse order, so that would suggest that any sequence-specific binding proteins will no longer bind. And then what we can do is, looking at the reverse complement orientation and the correct orientation, when you look at the overall structure, you see it's very nicely correlated, in which when flipping the sequence, you don't really disrupt the overall structure. And then I highlighted the 4 3' UTRs I'm using in this assay, which again are very nicely correlated in both the correct and reverse complement orientation. And we're very excited to see that when we inserted these in the reverse complement orientation, again, G3P1 is responsible for regulating those highly structured UTRs even though they're in the reverse complement orientation. The next thing is we wanted to further analyze one of these UTRs, so I chose the EF3B 3' UTR, and when you look at the overall structure, it kind of segmented it into these 3 different segments, in which it seemed like maybe one of these structures was responsible. Maybe it's just this one structure that G3P1 is regulating. So what I again did, I cut this 3' UTR up into individual pieces, inserted it into the 3' UTR as the only part of the UTR, and then stuck it into the system. And we're very excited to see that when G3P1 was knocked out, every single fragment was upregulated. So that's suggesting that any one of these pieces was sufficient to facilitate this interaction. So there is not, unless each fragment has the same structure, it doesn't seem to be due to a very specific structure, and instead it just needs overall structure to facilitate this interaction. And the reason we think it's due to the overall structure is that when we fold these individually, you can, based on the values, anything greater than 0.3 is highly structured, and all but one of them would be considered highly structured, and this one is almost highly structured. So that's kind of why we think this might be going on, is that even these individual pieces can be highly structured on their own. And so what we could now do is, what's really nice is we now have a much smaller fragment to work with. We have this little 88 nucleotide fragment that is able to be regulated by G3P1. So what we could then ask is, can we somehow disrupt this regulation? So what I was able to do is artificially design a sequence that forms very little structure, as shown here, which when G3P1 is knocked out, it has no effect, as we would expect. And what I can now do is take this unstructured sequence that's about 200 nucleotides long, and either insert it into the full-length EIF3B or that 88 nucleotide sequence to see what happens. So when we insert it with the full-length sequence, which is about 600 nucleotides long, you can first see that the overall structure is still highly structured. And still being highly structured, as you would expect, you see G3P1 level, when G3P1 is knocked out, it's still upregulated. So this is suggesting that this unstructured sequence is not inhibiting the ability for G3P1 to regulate it. However, we were very excited to see that when we actually insert it with an 88 nucleotide fragment in which you greatly reduce the overall structure, we saw reduction in the ability for GBP1 to regulate it. So somehow by reducing the overall structure of the entire 3' UTR, you can reduce the ability for GBP1 to regulate it. Oh. And then just to take a quick detour, or just to kind of reiterate the findings, everything I've been showing has been with GBP1 knockout. And one thing we just want to do is confirm it with the helicase-dependent UPF1. So this is looking at wild-type UPF1 in white, and then that helicase mutant in gray. So when you mutate that helicase domain, you see that UPF1 is no longer able to regulate the highly structured versions of this transcript, but when it's either unstructured or very poorly structured, you don't see the ability for UPF1 to regulate it when it doesn't have the helicase activity. So just kind of concluding these 3 points for this section is we were able to show that it was sequence independent through that reverse complement assay, and that in terms of minimal structures required is that we don't really have an answer for that yet because every single fragment from EF3B was sufficient for this regulation. And then in terms of the non-structured sequences, it seems like the unstructured sequences don't inhibit the interaction, but it can dilute the overall structure, which is inhibitory. So it's essential to have sufficient overall structure to facilitate this degradation. So then lastly, everything I've been talking about has been this overall structure. It doesn't really mean a lot probably to many of you. So I kind of wanted to have a way to better define what overall structure really means. My sister's laughing at me. So I kind of want to take a step back and further characterize what RNA structure really is. And to do that, we needed to use a few other metrics in terms of what RNA structure is. And one way to do that is using prior predictive programs that can give you the probability for an individual nucleotide being base paired or not within the 3' UTR. So based on the heat map, is when anything is more in the green and red range, these are nucleotides that whenever you fold it, it tends to be base paired with another nucleotide. Or anything in pink, normally when you fold it, it tends not to be in a hairpin. So that's why you can see that when you have these big hairpin loops, there's a very low probability that they're base pairing. But when you get into some of these hairpins, you see there's very high probabilities that they're base pairing. So what's really nice with this is you can kind of now look at the single nucleotide level, and you can also compare this to the overall structure. So when you look at the, based on overall structure, these highly structured UTRs, you can see compared to all, you see the shift to the right, which again means the highly structured UTRs have a lot more base pairing going on within the 3' UTR, and then the poorly structured UTRs are shifting to the left. So that's suggesting they have a lot less base pairing going on. So now because we have this single nucleotide level, is we can kind of look within the ClipSeq peaks to see where UPF1 and G3P1 bind. Do they tend to be bound in regions of base paired regions, or are they bound to single-stranded regions? So when you look, first just looking at all in black versus where the actual peak, is you see, again, a shift to the right. So this is suggesting that when UPF1 and G3P1 bound together, they tend to be in regions that are much more base paired. And then from the middle of the peak, we can expand it from the peak, and look at even surrounding the peak, that within the 50, 100 nucleotide, 200 nucleotide region, you still see that shift. So in the region it's at, it's even more base paired throughout. So to kind of better explore this base pairing phenomenon, is this responsible, is I could take that little 88 nucleotide fragment I've been talking about earlier and do some more manipulations to it to disrupt the base pairing. So when you have the full 88 nucleotide wild type, you see very high levels of base pairing. But when you delete one of the hairpins, you can drastically reduce it. Or what you can do is, when you look over here, is if you mutate one side of the hairpin, you can disrupt RNA-based pairing, which is then confirmed through the predictive programs. And lastly, what then you can do is you can restore the base pairing by mutating the other side of the hairpin to re-allow for base pairing. And we were very excited to see that we were able to see the same findings, in which only when you have the high levels of base pairing with the full-length 88 nucleotide, or when you correct that hairpin, can you see this upregulation by GBP1? But if you delete the hairpin or mutate the hairpin, you really reduce the ability for GBP1 to regulate this transcript. So this is suggesting that these hairpins are essential for GBP1 to facilitate the degradation of them. So everything I've been talking about has been using predictive programs to determine if it's base paired or not. I wanted to know, if you look actually within a cell, can you observe the regions to be base paired? And there are some nice, again, publicly available data sets in which they can look genome-wide to see at the single nucleotide level, is there base pairing going on or not? And so first, just reintroducing you to the predictive program. This is just looking at that individual hairpin that have been mutating and disrupting the function. You can see this is based on predicted programs what the folding is. But if you incorporate the various data sets, either the IC shape or the DMS map seq, you can hopefully appreciate that all three of these structures look very similar. So even when you incorporate data that's found within cells, which you can see is that when you have less than 0.4, the black nucleotides, those are the ones that have very low reactivity, which is suggesting they are in structures. That even when you incorporate this data of within cells, how structured they are, you can see this hairpin is still forming even in cells. And then we can then also ask, besides this little 88 nucleotide fragment, is this happening genome-wide? And so we can go back to those clip-seq peaks to where G3BP1 and UPF1 are overlapping. And because it's a DMS reactivity, everything's kind of flipped, so everything on the right is less structured and everything on the left is more structured. And you can hopefully appreciate when you look at all in black versus the peak in purple, you see that shift to the left in which where G3BP1 and UPF1 are binding, there's a lot less reactivity, which suggests there's a lot more structure in that area. I'm sorry. And then also when you expand within that region to 200 nucleotides, you still see that it has poor ability to react to DMS, suggesting more structure. So I kind of want to conclude with that, is that hopefully I've convinced you that UPF1 and G3BP1 senses overall RNA structure to regulate RNA decay. And that in this section, what I've really shown, hopefully, is that it's defined by RNA-based pairing density, and that these base pairings are confirmed through experiments, and that this is not through specific linear sequences. And some of the future directions I want to work on is determining the molecular mechanism of the degradation. I've talked about earlier that G3BP1 might be an endoribonuclease involved. That could be the endoribonuclease involved, so that's something that needs to be further investigated. And also looking into everything I've talked about, I don't really know what the biology is behind it, but there are some avenues that would be of interest to follow up. And if anyone's interested in these, I'm more than happy to talk about them. But I did want to just take a minute to talk about the circular RNAs, since some of you have been here earlier and known that was actually my initial project. So just to give a brief talk about the circular RNAs, is so these circular RNAs come from POL2's transcripts, the same place the mRNAs come from. And normally what happens is while it's being processed, it's processed into its normal linear RNA isoform. However, sometimes within these introns, you get some base pairing going on within the introns, which can then bring exons in close proximity. In this example, exon 5 is very close to exon 3. And what can happen is splicing can occur to connect these two together, which then produces a circular RNA. And what's very interesting about these circular RNAs is because they don't have a 5' or 3' end, they're resistant to other forms of RNA degradation, and these would require an endoribonuclease to regulate them. So because these would require an endoribonuclease to regulate them, we were very interested to see, can our pathway regulate them, which would suggest that our pathway might depend on an endoribonuclease. So what we're able to do is, again, my undergraduate Claire was able to help extract some sequences, and we can fold them. And when you look at overall how structured they are, you can see there's a very large population of circular RNAs that we considered highly structured. And in this one, we screened 37 different circular RNAs with varying degrees of structure, with the right being more structured, the left being less structured. And when you knock down UPF1 or G3BP1 in the DLD1 cell lines, as you can see, only when you start getting into these more structured circular RNAs can you find the upregulation, and UPF1 and G3BP1 have no effect on those poorly structured UTI, or the circular RNAs. But I just wanted to kind of briefly talk about this, because if it wasn't for these circular RNAs, there's no way I would have been able to find this pathway, and sometimes going through different avenues, you really change the direction of your research. So it's very useful to use maybe some less interesting RNAs that I'm guessing most of you have not heard about previously, to maybe get into some biology that more of you are interested in, since mRNAs are essential for making the protein, so they're a little more important and easier to talk about and find interest in. But just to conclude, hopefully I've shown you, based on everything, that we're able to identify UPF1 and G3BP1 are these two essential proteins to regulate the decay of the highly structured RNAs, and that it's through the decay and not biogenesis, and that it's this overall structure that's required through higher density of base pairing. And so with that, I'd like to thank the lab, as well as some of our collaborators, Rachel Green and Jeremy Willis, for some of the help they've been able to provide. My thesis committee has been great for really helping me out. They really challenged me, always ask for more and more and more. They really helped me out, made sure I really expanded everything, and they've been a great, tremendous help. Also the B&B department, which Anthony's lab is focused in, has been a great help, and they've really taken me in. The human genetics program, Dr. Valley and Sandy for all their help and support, and then just some of the funding sources that have really helped this project continue on. And with that, I'm happy to take any questions.
Questions for Joe?
So I'm getting caught up on the sequence independence of the structure. Is it just that, like, with a single transcript, for example, you have so many different options of base pairing that it doesn't really, the exact sequence doesn't matter?
Right, yes, and since all the genes being regulated, they all have different sequences, and there's no, you can actually use programs to look for motifs that are enriched. And Veronica, one of the members of the lab, she was able to look at that, and there aren't really any motifs that you find. There are no specific sequences that G3P1 is binding to. And that's why I did some assays where I could mutate the hairpin to change the sequence, but not the hairpin, and it was still regulated. So it's suggesting that it wasn't due to the sequence, but the actual hairpin itself.
One more follow-up. So we see with, like, codon, like, reading the gene code, that there's, like, wobble pairing, not the base pair, right? Does that play a role in, like, the structure or structural folding?
Well, so in terms of folding, since there's usually the A's and the U's base pairing and the G's and the C's base pairing, but with RNA, the G's and the U's can also base pair. So you have that additional type of base pairing going on. And then the other thing I can't say anything about is then these secondary structures will form a tertiary structure, which could be really what G3P1 is recognizing, but there aren't really any good ways to visualize the tertiary structure at this point. So we're stuck with looking at the secondary structure, which seems to be a good proxy, at least for the time being.
So, Joe, you listed off several mechanisms for regulation of RNA, and you also showed us that there are several genes which use this mechanism that you have described. So if you look across that collection of transcripts that are using this mechanism, is there any sort of biological connection? Is there any rhyme or reason why which transcripts use which mechanisms and cell-specific expression and stuff like that?
Yeah, so we've had looked into that one. Too specific, but one thing that's interesting is we're actually seeing a lot of metabolism genes that are being upregulated, or that have the highly structured DTRs. So it could have specific features in terms of metabolism. It's a lot of mitochondrial metabolism genes, so that's something that will be of interest to look into.
And what about the time course? In other words, in intact cells, is this a kind of regulation that happens over a very fast time course, or is it slower, or what?
Yeah, that's an excellent question. So if you think about nonsense BJK, which is much more well studied, that is extremely rapid, happens pretty much the second translation starts, so that's a very rapid pathway. For us, in these normal conditions, it's not the most rapid pathway. When I did those actinomycin D studies, I see about a 50% increase in the half-life. With nonsense BJK, I see about a 10,000% increase. So what we think is going on is that under normal conditions, it is happening, but what we think is most likely is there are some signals that will most likely either really activate this pathway or shut it off. So that are some things we're interested in hopefully discovering.
And is the expression of the UPF1 and the other one, gene, whatever it is, are they pretty much co-regulated when you look across different cell types, or are they?
They're both pretty highly expressed proteins, and then when you look, there's various mass spec studies that will try to quantify the numbers, and both of them are very high across many different cell types. So there's nothing really with that. But what is interesting in terms of co-regulation is that both of them have highly structured ETR, so there is a chance that there's autoregulatory regulations going on to try and modulate the levels of the two proteins through the mechanism.
Do you think other proteins are involved?
Yeah, we think most likely. I didn't go into it, but the G3P2, that homolog, is also involved in this pathway. Is that when they knock that one out, you do see the upregulation as well. And we assume there's other proteins involved. G3P1, like I said, is phosphorylated. UPF1 has a phosphorylation site that's important, so we're assuming there's some kinase probably involved in this pathway as well.
And are there any Mendelian phenotypes associated with these genes?
No. So if you don't have UPF1 and G3P1 are both essential proteins. So UPF1, you can't even have a cancer cell line without UPF1. So if you knock it out, they die. And people have tried to make G3P1 knockout mice, and those are also embryonic lethal. So no one's identified any mutations, and that might be because it's essential for development.
If you look at those four mechanisms, or it was, how many, where would this mechanism stand? Would it be one of the more important? Would it be lesser? What?
The most.
Yeah, the most. That's a good question. So as of right now,
What fraction of RNAs, you know, are using this?
right,
this
so
mechanism?
So in terms of genes being highly structured, we think there's about a third of them that might be considered highly structured. RNA sequencing didn't pick up all of those, but we did pick up a large fraction of highly structured ones that are upregulated. So it does have a high capability. And then the other thing is nonsense BTK is pretty specific to bad transcripts that have those premature termination codons. So it has a very specific function for regulating those. We don't know why GPP1 and UPF1 want to regulate these transcripts. We need to figure that out still to get a better idea of why this is important. So far we know it's there, so now the next question is why. And some of those biological questions, such as like RNA viruses, might be a good way to understand why this is important.
How about evolution?
That's
I
a
mean,
good question. Yeah.
where does it stand in?
So UPF1 and GPP1 you can find in yeast. They have homologs in yeast. But then the problem is you need to have a 3' UTI that can form the structure to be regulated. And when you go to mice, you see there is actually a lot of conservation between them. I think I have that. I can show you to make it easier. Is when you look at the genes in which is homologs between the two, you can look at the human overall structure to the mouse overall structure, and you can see they're very nicely correlated. So there is conservation in overall structure. And then what you can do is to make sure it's not just due to sequence conservation, is you can align the two sequences. And it's a little convoluted to look at the time, but basically is this is looking at the change in the overall structure compared to the sequence alignment. So when you look at the population up here, these have the same overall structure because they have the same sequence. But when you look down here, these are the genes that have very different sequences but the same overall structure. So this could be a mechanism they conserve the RNA structure to be regulated by my proteins, but they change the sequence completely and they adapted and changed to maybe change the sequence-specific RNA binding